It's a distinct pleasure for me to introduce today's Grand Round speaker, Dr. Leena Hilakivi-Clarke, who is coming here via Zoom from Hormel Institute. Dr. Hilakivi-Clarke received her PhD degree from Helsinki University in Finland in the field of experimental psychology and physiology. And subsequently, she received a Fogarty postdoctoral fellowship award at the National Institutes of Health in Bethesda. And so she spent 3 years there. At the end of that time, she was offered a position at the Georgetown University, and she progressed at Georgetown University from assistant professor to full professor, and in year 2020 she moved to Hormel Institute as assistant director of research and professor of nutrition and food science. Dr. Hilakivi-Clarke has a broad range of interests in the context of breast cancer. She has been involved in study of nutrition, the effect of nutrition, the effect of obesity, microbiota, stress, and immune microenvironment, and so a whole variety of areas in relating to breast cancer, where she has made a significant contribution over the years. One of her publications received the in 2017 received the Society for Endocrinology Journal Award. So we are delighted that Leena Hilakivi-Clarke has been kind enough to accept our invitation to present today's talk dealing with diet, gut microbiota, and response to cancer immunotherapy. Leena, please go ahead.
Thank you very much for the great invitation, and I was particularly impressed the way you pronounced my name. It was perfect. So I'm going to talk to you today about diet and its impact on gut microbiota and response to cancer immunotherapy. So we know that the immune checkpoint inhibitors or blockers have revolutionized the treatment of many cancers, and they are now a standard of treatment for advanced melanomas, non-small cell lung cancer, and several other cancers, including triple-negative breast cancer. However, only 20 to 30% of the treated patients will respond to the therapy, and some of the responding patients then become resistant to the therapy. And there are several factors that seem to be impacting the primary and the secondary resistance. And those factors include both tumor intrinsic and factors arising from the complex interplay between cancer and its microenvironment. In 2015, a paper was published in Science, and that was a study done in mouse, showing that mice coming from different vendors responded completely differently to immune checkpoint blockage therapy. And then 3 years later, there were 3 papers also published in Science that showed that in humans, the gut microbiota was impacting the responsiveness to how melanoma responded or lung cancer responded to immune checkpoint blockers. And here is a summary of the human studies. So the non-responders, the gut microbiota contained increased abundance of bacteriocytes. And then in the responders, there was an increase in Bifidobacterium, Akkermansia municipala, and Faecalibacterium. The problem was that in each of these human studies, a different set of bacteria seemed to be predicting the responsiveness, but common to them was that the bacteriocytes were elevated in non-responders. And then the investigators took samples from the humans and put them into mice, and found that indeed the mice that got gut microbiota from non-responder wasn't able to respond to immune checkpoint blockers and the tumor grew, whilst the mouse that got gut microbiota from a responder human, they responded well. And they also found that the responder mice had an increase in CD8 T cells, and the non-responders had an increase in T regulatory cells. So now this was a very nice review paper about the anti-PD-1, and unfortunately I can't see my topic very well because of this thing at the... Can you see when I'm moving the thing, or no? I just moving it to myself. So anti-PD-1 in the world of gut wonderland. So there are factors and I don't know whether my screen froze. Okay, there are factors that are linked to favorable gut microbiome or favorable response. So Akkermansia municipala and Ruminococci and Bifidobacterium, different species have been linked to favorable outcome, and then high alpha diversity. And the unfavorable gut microbiome includes low alpha diversity and particularly Prevotella in the Bacteroidetes phylum. Antibiotics seem to be having adverse effects on the response, but it's not clear when the antibiotics have to be consumed to have harmful effects. So it seems like it's a year, 2 years before the therapy starts that it could still have an adverse impact. And you probably all know what the gut microbiota is, but I just want to very quickly highlight that the human body is composed half and half of human cells and bacterial cells. But of the genes that are generated by the cells in our body, only 1% are expressed in human cells and 99% are in the microbes. And there are several ways, or two main ways, to study the gut microbiota composition: 16R RNA sequencing, or then the Shortchain sequencing. And it has been assessed what kind of, so you saw here quickly that there are, or you can see it very well, that there are several phyla, family, genus, and species, but there seem to be 6 major phyla: Firmicutes, Bacteroidetes, Actinobacteria, Verruco microglia, Proteobacteria, and Tenericutes, and most of our gut microbiota composes of the Bacteroidetes and the Firmicutes. And when we look at the literature and wonder what specific genus might be important, it's the Lactobacillus bifidobacterium and Akkermansia. When they are elevated, that seems to be linked to good health and good outcome to immunocheckpoint blockers. I just want to point out that recently 4 of the main phyla have been renamed, and like I was confused what is Bacillota, so that's Firmicosites. The other ones are more similar to their older names, and I don't know how widely these new names are being used, because there's some controversy about this naming and whether it's justified or no. So I just use the old ways. The gut microbiota is in our body, or microbiota in general, because it serves several different important functions. It has a metabolic role. It helps digest the foods we consume. It has a barrier role, so it protects the human cells from the toxins that we might consume through the food or what we drink. It is also very important in regulating the appropriate immune responses. It's the largest immune organ in our body, and it has a maintenance role, affects the maturation of the gastrointestinal tract, and provides food for all our cells. The bacteria itself is important, but most important are the metabolites the bacteria generate. And this nice paper published in Nature Reviews summarizes lots of the important metabolites of the gut microbiota. I just want to point out the when we consume fiber, that increases the enzymes, and that leads to an increase in short-chain fatty acids. And these short-chain fatty acids have multiple roles in the body. They provide nourishment for the epithelial cells and can have then their pro-inflammatory role, affect food intake and energy expenditure. In the immune cells, they have an immunomodulatory role, particularly through the short-chain fatty acid receptors, and they also impact adipocytes, and they act as ASTAC inhibitors, so they can modulate the epigenome. So now, when we know that the gut microbiota is important in regulating responsiveness to immune checkpoint blockers, we can ask, how do we modulate it to get the best outcome? So there has been lots of interest in probiotics, but they are also quite controversial. So diet seems to be a much better way to impact the gut microbiota, and by consuming probiotic foods or prebiotic foods. So the probiotic foods are foods that contain active bacteria, and there are some very nice foods to consume, like yogurt, bitter chocolate, aged cheese, that will contain these active bacteria and can modify our gut microbiota. Prebiotics are the foods that are high in soluble fiber, and one of the highest sources of prebiotics is onion, asparagus, and garlic, artichoke. Bananas have them, and there are also several different prebiotic tablets that one can consume, and I just put here that it was very difficult for probiotics. I found a picture where there is no specific company that produces, but prebiotics I couldn't. So I just say I'm not supporting any of these lines. They are just to show that there are several of them available. So then I'm gonna review 2 studies that have been looking diet and gut microbiota. The first study, and that was published in Cell. And this is just looking how the diet can modulate immune system. And in that study, and so not yet looking responsiveness to immune checkpoint blockers, but I think this is a very interesting study and worth going through. And after I go through these 2 studies, I will share with you the study that is ongoing in my lab. It is really ongoing, so I would appreciate if you have any insight or any suggestions for the study, but we talk about it when we get there. So in this human study, there were 381 people that were eligible for the study, but most of them had to be excluded for several reasons. So then the investigators selected 39 people, and they allocated them either to individuals who were in the high fiber diet or those who got high fermented foods. And of the high fiber diet, 3 dropped out, and none in the fermented food group. And what the investigators asked the people to do was to increase, in the fiber group, was to increase their, I don't know why my slides seem to be stuck. Okay, here. So they were asked to, in the fiber group, they were asked to consume these foods to increase the fiber intake by 20 grams a day. And in the fermented food group, they were asked to consume these foods, cottage cheese, kefir, kombucha, or other drinks that had the live bacteria, vegetable brine drinks. Those are very high in the probiotics, vegetables, or yogurt. And they were asked to consume 6 or more servings per day. And the investigators then studied at the baseline, did the food log, and did the gut microbiota assessment also. So the bacteria, the metabolites, proteomics, and then assessed short-chain fatty acids. And then they studied inflammatory cytokines. And the ramp period was when the individuals were hoped to, that they changed their diet the way they were asked to. There was a maintenance period, and then there was given a choice period. And the people in the study were very good participants because they increased their fiber intake from 20 to 40, which was as the investigators hoped for. In the fermented food group, they didn't get to 6 servings, but 5 servings, which is quite good. So the intervention was successful. Then when they looked the outcomes of the study in the fiber, they found that the enzymes were increased, but short-chain fatty acids were not altered very clearly, at least. In the fermented food groups, they found that basically all the inflammatory markers they looked were reduced. So the summary of this study is that high fiber diet leads to an increase microbiome function, particularly increase in enzymes, which is great. And there's a little bit of a personalized immune response. In the high fermented food group, there was a not specific change on the gut microbiota, but there was an increase in microbiome diversity, which is considered to be a healthy outcome. And then there was a decrease in inflammatory signals and activity. The other study that was published late last year, and I think it came actually out early 2020 this year, was to look dietary fiber and probiotics, their influence on gut microbiome and response to immunotherapy amongst melanoma patients. And in this study, there were several populations. So these individuals were not asked, patients were not asked to consume any particular foods. They just were part of a study that patients that received immunotherapy, but they were assessed for their food intake and their intake of probiotics. And then there was a mouse study, and the mouse got either the probiotics or then fiber. And when they looked the humans, they found that the individuals who responded to immune checkpoint blockers, anti-PD-1 therapy, had increased levels of ruminococci and Bacillobacterium, and that was significant. And then they looked how it related probably their diet. So if there were some individuals, not many, but, and I can't remember now whether it was 17 out of 40 and 49 didn't take probiotics, and they were all kinds of, so the investigators didn't assess what particular probiotics contributed to this outcome, but they found that if they didn't take, they had better survival than if they took in amongst the melanoma patients. And then they did a study in mice where they took gut microbiota from a person who responded really well to the immune checkpoint blockers and put that to germ-free mice, and then asked the mice when they had taken the donor gut microbiota, asked them with probiotics, and then looked responsiveness to, then they xenografted them with the melanoma cells, human melanoma cells. No, actually, I think this is syngeneic mice, so mouse melanoma cells, and then looked the responsiveness to anti-PD-L1 therapy. And they found that the controls responded really well to the therapy, the anti-PD-L1 therapy, but if they got lactobacillus-based or bifidobacterium-based probiotic supplementation, the responsiveness was much more poor. And when they looked at immune cell responses, there was nothing there to report, basically. And then they looked at dietary fiber intake and response to anti-PD-1 and found that those who had sufficient fiber intake had a better survival from the cancer than those who have insufficient fiber intake. And they were able also to divide their participants to those that only the red line, I don't know what happened, okay, is the one who only took who only had sufficient levels of fiber, and the 3 other groups were those that had insufficient fiber plus probiotics or probiotics only. So clearly, just consuming enough fiber was beneficial for the patient survival probability when the patients were treated with immune checkpoint blockers. And in the study they did in mice, so again they they treated them with fibers and look the response to the anti-PD-1 therapy in this case. And here, the lowest line here, the tumors that didn't grow, are the ones that get the anti-PD-1 and are consuming high fiber, whilst the others are those in low fiber or not getting the therapy. So dietary fiber improves melanoma response to immune checkpoint blockers in humans, and dietary fiber improves response to immune checkpoint blockers in mice. And they, in this part of the study, they just characterized the signaling in the tumor infiltrating cells and found that the immune pathways were, so these were the individuals that didn't got any immune checkpoint blockers, so they are pretty similar to each other, and they got, it didn't matter if they got fiber or no, or if they were consuming fiber or no, whilst if they were consuming lots of fiber and got the anti-PD-1, then the signaling pattern of the immune cells was quite different. Okay, then I'm gonna talk to you about the study that we are doing at the moment. And we started the study at Shortchain, so a couple of years ago, and this was before either of the studies I just talked about were yet published. So what we wanted to, and here I just want to highlight 2 investigators at the Hormel Institute who are working on this project, Fabia Andrade and then Vivek Verma, who they both come from Shortchain, and Vivek recently was appointed as an assistant professor at Hormel Institute. And so we wondered if the lab diet that was given to the animals might impact how they respond to immune checkpoint blockers. And here we used breast cancer cells or mammary tumors. And so when the animals are given the regular lab diet, this one is 50-58, that is the primary ingredient is whole wheat, and it has several other compounds too, and it has the same level of protein and fat, and its energy density is similar to the other diets, but what it has tons of isoflavones, and the isoflavone content varies from batch to batch, and the level varies from, it's on average about like Asian person consuming 2 servings of soya foods daily. So the one diet that we used was this 5v5M diet that is also whole wheat based, but it has the same amount of isoflavones from batch to batch. And this is quite low. This is mimicking what the Western people consume when they consume soya foods, or when, for example, soya flour has been added to the bread that they consume. And then we often use this AIN93G diet because it's a semi-purified diet. Corn sugar is the primary ingredient, and it has no isoflavones, so it's well controlled. So in addition to having these 3 different diets, we also wondered if because we had breast cancer cells that we're looking at, whether the estrogen receptor might be playing a role in the responsiveness to the immune checkpoint blockers. So, and we used both triple negative breast cancer cell lines, so they don't have the estrogen receptor, but the immune cells do have estrogen receptors. And when the estrogen receptor is activated in the immune cells, that promotes immunosuppression and resistance to the immune checkpoint blockers, because it activates the myeloid-derived suppressor cells. So if one blocks the estrogen receptors, that could then help perhaps the responsiveness to anti-PD-1 therapy. And in fact, there was a study published in iScience after we started doing our study and got funding, R21 funding, to do some additional studies, which looked at this same question, but specifically giving the animals ER-beta antibody. So they activated ER-beta because when ER-beta is activated, it stimulates the CD8 cells and suppresses the myeloid-derived suppressor cells. And they used 41 breast cancer cell line. This is triple negative breast cancer. And they found that when they just gave, these cells are not particularly responsive to immune checkpoint blockers. As you can see here, that there was some reduction with the anti-PD-1. In the autotopic model, not so much. The ER-beta agonist also did slightly reduce tumor growth, but not significantly. But when the animals were given a combination, that was highly effective in reducing the tumor growth. So genistein is something that is found in soya foods particularly, and so isoflavone genistein, and it's known to modify the gut microbiota. The problem is, in terms of breast cancer, that when we look at the chemical structure of genistein, it's very similar to that of the estradiol. And in fact, genistein only, as far as we know, binds to the estrogen receptors, but it binds preferentially to ER-beta, so the one that in the previous study was used to stimulate to get the response to immune checkpoint blockers, and then it also binds and activates GPAR. And very briefly, we did a study that was published in Nutrients last year, where we first modulated the responsiveness of the animals to anti-estrogen therapy by exposing them during fetal life to a high-fat, obesity-inducing high-fat diet or control diet. And when they are born to dams that are obese, they have an increased incidence of getting mammary tumors. They have increased incidence of mammary tumors, and we initiated the mammary tumors with the carcinogen DMPA, and we used rats. The reason is that when we, the rats, when they get DMPA, they get ER-positive breast cancers that will actually respond to the anti-estrogen therapy. So amongst the control animals, they respond pretty well to the therapy, but if they are born to an obese mother, they show increased resistance to the tamoxifen treatment, and they show increased threefold higher reoccurrence after the tamoxifen is taken out. And in this study, we supplemented the animals with genistein when they showed a response to tamoxifen, and we found that when we supplemented, that significantly reduced their risk of reoccurrence. In the control animals, the genistein supplementation didn't have any adverse effects. And when we looked at the gut microbiota of the non-supplemented animals, they had reduced levels of Clostridia that produces short-chain fatty acids, and increased levels of these Prevotella and Enterobacteria that are inflammatory. And genistein supplementation reversed all these changes. And here are some bacteria that also were significantly altered by the genistein supplementation. Verrucomicrobia was increased, and Akkermansia municipala was increased, which are the bacteria that predict good response to immune checkpoint blockers, and Streptococci was reduced. And we also found several metabolites that were altered by genistein supplementation, chloretine, ribothiamine, and some of them, and we also found more estradiol in the fecal samples in the genistein supplemented animals. And some of the changes were similar regardless of the maternal diet, and some were opposite. So this is just basically to show that the genistein supplementation will affect the gut microbiota. So then we did this study in mice where we fed them the 3 different diets: high genistein, control genistein, and no genistein. And we allografted them with the triple negative EO771 cells to mammary fat pad, and then started treating them. We implanted them with tamoxifen pellet or put the control pellet, and then treated them with the anti-PD-1 or IgG as a control. So before we go to see what happened to the response to the these tumors by the tamoxifen and immune checkpoint blocker, I just want to show an ongoing study where we looked if these treatments actually affect the EO771 cells directly. So there's no immune cells here, and these were luciferase marked cells, so we have using them also when we allograft them. And the red line here is the vehicle, and when the cells were treated with genistein 1 micromolar, which is a person who consumes 1 serving of soya foods a daily gets this amount. If they cells were treated with 5 micromolar, that is Asian person consuming regularly a couple of servings of soya foods a day. So it seemed like these triple negative breast cancer cell lines responded favorably to this genistein, whilst they didn't, this higher dose of genistein, whilst they didn't respond to the lower dose. And when we treated them with tamoxifen, it didn't have any effect on these cells, which is what one would expect, because they are triple negative. But when we treated them with fulvestrant, there was a response. And I just want to point out that fulvestrant So tamoxifen doesn't have any impact on IR-beta, but fulvestrant has been reported to increase the IR-beta. But it's a somewhat controversial whether it does or doesn't, and protein and messenger RNA levels data is somewhat different. When the cells were treated with the combination of tamoxifen and low-dose genistein, that adversely affects the cells, and perhaps slightly so also the fulvestrant treatment. However, the animals that got, not animals, cells that were treated with fulvestrant plus the high dose of genistein or tamoxifen and high dose of genistein stopped growing very nicely. And this is just to show, point out that the triple negative breast cancer cells, some of them are also IR-beta positive and can also have the G-bearer. So this is when we treated the animals with the diets, the 50-58 diet, the high in genistein and perhaps high in soluble fiber. We found that we gave them a non-responsive dose of anti-PD-1. The EO771 cells are very responsive to anti-PD-1, so we gave them half of a dose that normally is used. And we found that, yes, it didn't affect the tumor volume, but when tamoxifen and PD-1 were given together, there was a very nice response. So one can say that there was tamoxifen improved the responsiveness when there was lots of genistein and perhaps fiber in the diet, but the genistein may have also blocked the effect of anti-PD-1. And why I say that is because when we give less genistein, then we see that the anti-PD-1 alone was equally effective as the combination treatment. And if there was no genistein, then PD-1 was most effective, and the combination wasn't. It was still reducing the tumor growth more than the tamoxifen itself, that was completely ineffective. But so the combination wasn't as effective as it was when some genistein was in the diet. And when we looked at the anti-PD-1 response, one can see that it was worse in the animals that got the diet containing genistein, and the combination response was best if there was genistein in the diet. So why? And we also looked if the combination treatment or anti-PD-1 would affect ER-positive breast cancers, and we used the DMPA model, and we used mice here because rats, we are not aware of antibody anti-PD-1 that we could give to them. And here you can see that it didn't matter what diet they got, they didn't respond either to PD-1 or tamoxifen plus PD-1, and they were completely unresponsive to tamoxifen alone, which is what the mice are, even though they have the ER alpha receptor. And in the ER-positive model, we still saw that there was a better response to PD-1 when the animals didn't have any genistein in the diet, even though it wasn't, the responsiveness to anti-PD-1 wasn't significant. So now at the Hormel Institute, we have started to do this. So these studies were done all when we were still at Shortchain. So Hormel, we have started to do these studies, and instead of using different diets, we give the animals all the AN93G diet, and then we add 500 ppm genistein that mimics, generates perhaps 2-3 micromolar concentrations of genistein in the circulation of the animals. And when they got the genistein in the diet, there was a little better response with the combination. And here we also used fulvestrant instead of tamoxifen. Fulvestrant is more effective in inhibiting the estrogen receptors, whilst tamoxifen is a baso agonist and antagonist. And so the conclusion from this study was that the fulvestrant plus anti-PD-1 combination was significantly more effective in inhibiting the E077 mammary tumors than IGF control or fulvestrant alone, and the combination didn't clearly modify the response. And when there was no genistein in the diet, we again saw that the PD-1 was really effective in inhibiting the tumor growth. The combination didn't work. So it was somewhat similar that we got with the different diets. So anti-PD-1 inhibits tumor growth when no genistein is present, and fulvestrant also inhibits the growth of, which was kind of a different from the tamoxifen. So fulvestrant alone was effective if there was no genistein in inhibiting the cell growth. And so whether this is because the fulvestrant affects the immune cells, or whether the fulvestrant, as I showed you earlier, actually inhibits the growth of the tumors, we don't know yet. And here you can see when we compare the anti-PD-1 treatment with genistein or no genistein. So clearly genistein impairs it, but the combination therapy is somewhat different. And there it seems that when they get genistein in the diet, they respond slightly better. So now we have done the experiment third time, and here we put fluorescent in the E077 cells, and so we can see here the tumors growing in the IgG treated, and here smaller tumors in the PD-1 fulvestrant treated animals. And when the animals are getting genistein in the diet, they respond better to the combination treatment than PD-1 treatment. But here, for some reason, and this is what happens when we do these experiments, the PD-1 wasn't the most active, and fulvestrant actually stimulated the tumor growth. So do this data make any sense? So I try to summarize here. Anti-PD-1 monotherapy, when there's a complex fiber high genistein diet, it impairs response to anti-PD-1 therapy, and so does genistein alone. And when there is no inflammatory response to tumor microenvironment. And I forgot to tell you that we, in the first and third experiment, we were able to put the tumor cells without cutting the mammary fat pad, but in the middle experiment we had to cut it because when we started doing these studies at Hormel, we didn't have the same assistance as we had at the Shortchain, and when you cut the skin, you induce an inflammatory response. So whether that affected the data, and then diet that is devoid of complex fiber and genistein impairs response to anti-PD-1 when no local inflammatory response is induced. And the anti-PD-1 and anti-estrogen combination therapy, fulvestrant adds to the response to the immune checkpoint blocker, regardless whether genistein is in the diet or not, when there is no local inflammatory response. When local inflammatory response is induced, fulvestrant adds to the effectiveness of anti-PD-1, and tamoxifen also adds to the response when mice are fed diets containing complex fiber and genistein. So we have very quickly tried to start to look at the immune cell changes in these mouse studies, and so there is no change in the CD8 cells in the different treatments, animals in control diet or genistein diet. And when we look at the CD8 cell activation, there is so much noise that nothing is seen, and it's not so that there would be, so there is probably with fulvestrant, there is less activation if there's no genistein, but when there's genistein, it's all these treatments are slightly higher than this one, but it's not significant. Then when we look exhaustion, again, there is no difference. And I just out of curiosity, just eyeballed the different data to look the ratio between activation and exhaustion. And interestingly, the highest level of activation is when they just got the IgG, when there was no genistein present, but when there was genistein, it was about the same ratio in all of them. We have also done nanostring analysis from in the first study where we compared in the tumors that we harvested at the end of the therapy, which is not the ideal way to do it. We looked the signaling in the animals that got the combination versus the 3 other groups, and we found that the Th17 cell differentiation, those pathways were most affected, and there were kappa B signaling, and we have tried to look IL-17, and we were able to confirm some of these changes in the Th17 cell differentiation. That was most changes happened. We found confirmed, for example, ROR gamma and ROR alpha were activated with the combination treatment, and so was IRF4, and the NF-kappa B signaling pathways was reduced. We also saw some opposite results than we found in the nanostring analysis, and we have been looking other pathways too, which is like, for example, MYC seems to be significantly suppressed, and I tried to look if MYC is somehow related to response to immune checkpoint blockers, and I'm not sure, except that it seems to upregulate the PD-L1. So one would think that actually the upregulation would be good, but here we saw downregulation, and VCAM was also downregulated, and then GPER was significantly upregulated. So the conclusion, if one can draw any conclusion, is that factors that modify the gut microbiota impact responsiveness or refractoriness to cancer immune checkpoint blockage therapy. And diet is among the factors that affects the gut microbiota, but there are many other factors too, like physical activity, stress. So physical activity, beneficial effects, stress, adverse effects, exposure to endocrine disrupting chemicals and other environmental hazards negatively impact. So depending if the patient is coming from environment that don't have too much of these or has lots of them, could impact how they respond to the immune checkpoint blockers. And what dietary factors are beneficial and what are harmful remains to be determined. Beneficial might be soluble fiber, fermented foods, ketogenic diet, and short-chain fatty acids. Both of them, there are some studies, but one study in each type of dietary intervention. And then harmful, it may be that probiotics are not at all a good idea when one is getting immunotherapy. So I almost used all my time. So if you have any questions, and I stop to share so I can see you.
Yeah, no, thank you very much. Please go ahead and ask any questions you may have.
Thank you, Khalil. Well, that was a terrific talk, Leena, and a sauna anecdote, which you were talking about in the beginning. I have a son, daughter-in-law, and 2 children in Vasa, and they take saunas every day, and my son jumps in the Baltic,
Yes.
when it's freezing cold. So I'm quite familiar with it. Fascinating talk. I've got numerous questions. I may set up something separately to talk to you. I wonder, in doing the fiber studies in the humans, would it be more controllable to use some prescribed thing like Metamucil or something where you know the absolute amount of fiber, as opposed to relaying what individuals may relate about what they eat, because it may not be entirely accurate?
Yes, so the study where they gave the fiber, they didn't look at the immune checkpoint blockage response, so it was okay to give the diets. They just wanted to see what is modified, but I think it would probably, if one does a clinical trial, it probably is better to use the prebiotics form as a supplement. And there are several animal studies that have been done using these supplements, of course, and those suggest that there is a beneficial interaction.
Okay, thank you. Another question, if I may. I was intrigued by the high-fat diet and breast cancer examples you gave in the mice. And I wonder if you have looked at calorie restriction at all in this, which may feed into the ketogenic diet anecdote you mentioned. The reason why I mention that is a former faculty member here named Edmund Eunice, about 30 or more years ago, published a series of papers in mice where they looked at tumor uptake and growth in animals, and they put on a significant calorie restriction on the animals, and they lived like 20%, 25% longer. And I wonder if that's played into any of the studies you've thought about.
So my colleague at Shortchain looked at the maternal energy restriction, and she found that actually had a similar harmful effect as feeding them the high-fat diet. But there one animal study looked at ketogenic diet, the calorie restriction, and found that the responsiveness to immune checkpoint blockers was improved. Oh. And of course there are for example, the ketogenic diet is supposed to be anti-aging diet, and so is caloric restriction. So the data translated from mice to humans. But the thing is that quality of life for some people who love food may be impaired, so they might be so stressed out that it doesn't help them.
Yeah, and who doesn't like food, right? Thank you.
Any
Thank
other
you.
question?
I have a quick question. You were talking about earlier in the talk about the gut bacteria and the metabolites that they produce being important. So I'm curious, I know that there are gut bacteria that contribute to endocrine resistance, as well as gut bacteria that help with immune response. Are these completely distinct types of bacteria, or is there overlap?
I don't think actually nobody has looked if the gut microbiota contributes to endocrine resistance. And this is something that we really want to study. So there's an investigator at university and an investigator in Mayo who are very interested in taking a look in humans. They are breast cancer oncologists. And I'm planning to do an animal study with the DMPA model where we can study that. So, and we know that the chemotherapy, radiation therapy, all those are dependent on the gut microbiota, but nobody has looked at the endocrine therapy.
Thank you.
Is it possible to analyze the metabolites that are produced after a given diet?
Yes, so that would be a good idea to look them not only in the gut, but also in the circulation, like for example, the short chain fatty acids, because one can get them directly from the diet too.
And how rapidly does the microbiota change after changing a diet in an individual?
So that's debatable. Some studies don't find, even though they make the humans to consume long time, and some find in a couple of weeks. And so if we think our own experience, you eat something new and something that doesn't mix well with your existing gut microbiota, you feel it as a bloating or so the gut microbiota, I think it responds pretty quickly.
Thank you. Any other question? Well, if not, I'd like to thank you again for a superb talk. It's very knowledgeable and gave us a lot of new information that we were unfamiliar with. So thank you again for taking the time to give us a grand round talk today. Thank you.
Thank you very much.